So we talked about their reproduction. And I mentioned that they actually reproduce sexually and asexually, which is not uncommon, but it's definitely something that we don't always see in most phyla. Usually they're sexual or they're asexual. Usually they're not both. So some of the ways they can reproduce asexually and sexually, they can go through well, at least for asexual, they can go through a process of growing external buds. So we see here's a picture. It'd be easy to imagine that this is a growing sponge, and it starts to bud and grows a second sponge off of it. So that would be asexual reproduction. They can also produce a gemmule, which is where they bud, but it buds internally, and that bud is then detached, floats out, and drifts away, lands someplace else, and then will grow into a sponge. Because they're a loose connection of cells and there's no tissue or organs, if you break one of these in half, like break the top off of this sponge and it floats over and lands over here, it could actually attach and start to grow again because there's no systems inside of the sponge to be interconnected. So you're not killing anything really by detaching them, you're just detaching them. So fragmentation can definitely help them reproduce asexually. But aside from that, we do have some other things that they go through, like they can go through regeneration. And regeneration is just where if a part of a sponge is broken off, they can regrow it and reproduce that piece that is missing. I mentioned earlier that if you took a sponge and you put it in a blender, essentially, and you broke it up into its base components, that like loosely disorganized cells will start to reorganize themselves and rebuild a sponge. We call that somatic embryogenesis, meaning that the cells, because they're not reproductive cells, they're somatic cells, right? They're not sperm and egg, they're every other type of cell. Those somatic cells can actually reorganize into a functioning sponge, and that process is called somatic embryogenesis. Sponges are cool. That's all I can really say about that. I think that's really, really neat. All right, so looking more into their sexual reproduction, things can get a little bit crazy. They are monoecious, meaning that they're both male and female, so they're hermaphrodites. Essentially what happens, because they don't have a lot of different types of cells, so some of the cells that they do have will simply modify into sperm or into egg in order to reproduce sexually. So the choanocytes, the ones that already have the flagella and the little collar on them, they will modify themselves into sperm. The archaeocytes, the amoebocytes, will actually modify themselves into the ovum, into the egg. And they can just, they do all kinds of cool things. Some sponges release their sperm into the water on when the temperature is right, and the moon is right, and the tide is right, and hope to fertilize an egg. Sometimes the eggs are released into water. Sometimes the eggs are held on the inside. It just depends. So we have viviparous sponges, and in that case the sperm is released into the water, but the ovum, the egg, is not. So the egg stays on the inside, and as the sperms are filtered by another sponge and get inside of another sponge, they will fertilize and fertilize the egg. Essentially, it depends on the egg as it grows and develops and it's young. It will depend on the nutrients of the essentially the parent that it's in. We call that, we'll just say, the mother, and until it'll keep doing that until it's ready, and it'll break off and then float over and attach someplace else and continue to grow. So that's viviparous, essentially meaning live birth. Then we have oviparous, where the sperm and the ovum, or the egg, are both released into the water. It produces, they'll meet together in the water and go through conception and fertilization, and then it forms a free-floating larva called a parenchyma, which is a fun word to say, parenchyma, and that parenchyma will then attach to something and start to grow. So the viviparous and the oviparous is really where is the conception and fertilization happening. If the egg is not released, if it produces sperm and produces an egg, and the egg is held on the inside and not released, and the sperm get to it, its fertilization and conception is happening on the inside of a sperm, so it's considered to be viviparous, because then it would detach, break loose, and go out of the parent like a live birth. Whereas oviparous, both sperm and egg release floating around the water. They'll meet together. Conception and fertilization happens in free-floating water. They form a parenchyma. The parenchyma will attach to something and start to grow. Yay! They do all kinds of cool things. Like I said, sponges, they do any way they can. So a little bit about the peripheral development. Essentially, there's two different ways that they'll develop. Because they are a loose conglomeration of cells, they often don't have to go through the gastrulation process that we've looked at that most animals will go through. For example, in the oviparous one, the perichimula, when it attaches to the ground, it's just a loose blob of cells. The cells will start to organize themselves to form a cavity in the middle, form that spongiocele in the middle. They'll form some tubes going to the outside, those canals, and so forth and so on. And they don't really go through any type of it happens a little bit differently with the viviparous ones than it does with this. So this is for the oviparous. The oviparous would just blob and morph into a new sponge. There's no really pattern of development. Cleavage doesn't occur in any type of way. But with the viviparous ones, the egg will actually go through cleavage and start to divide. It'll form the blastula, which is the hollow ball of cells. That will then go through gastrulation and start forming the different canals and tubes that are on the inside of it. But again, this is just a loose connection of cells. There's no organs or systems for them to worry about. So essentially, once it just develops some tubes, it's good to go. The collared cells would then go where they essentially lodge in and form where they need to, so that way they'll start pumping the flow of water, and the sponge can start feeding. So they're different because they're so simple of a type of animal. That is often also reflected in their development.